screen. It should be okay now. All right, yeah, as long as everybody, there we go. Well, welcome everybody to an introduction to the Ministry of Agriculture Veterinary Diagnostic Lab. The BC Goat Association has organized this webinar with Dr. Glenna McGregor. So a few introductions, because we have quite a few people that signed up for the webinar, which is fantastic. I asked everybody to just do a little survey so we can kind of get to know you and you know, see where everybody is and what you have. And so far we have about 33% of you are from Vancouver Island and surrounding islands. And we've got about 22% from the South Coast, 16% from the Cariboo-Jacotan, and we have 11% from the Thompson-Nicola Valley, and 16% from the Kootenays. And we have, what do we have? About 55% have goats, and 11% have sheep, and we have about 11% who have both sheep and goats. And we have about 22% who haven't bought any sheep or goats yet. So that's exciting that we have some new people that are looking at getting goats and sheep. And we have quite a few that are just starting out in 1 to 2 years. And we have a few in 3 to 5 years. And then we jump right over to the 10 years. We have about 30% in the that have had animals over 10 years. And we've got quite a few. What do we have? About 21% have just started out, just have about 1 to 5 goats. And where do we have? About 15% with 5 to 10. We don't have anybody with over 100, but we do have one person with 50 to 100, which is great. And we've got quite a few mixtures, 15 to 20 and 20 to 30. Same with sheep. Most have quite a few. We've got about almost 80% have about, what is that? It's about 30 to 50, which is great. And what was okay? And what people would like to see for the next webinar would be management protocols. There was about 40% would like to see that. So that might be something that we'll look at for our next webinar. So that's very exciting. So a little bit of housekeeping. We are recording this webinar for public viewing in the future. We ask that you keep your mute on during the webinar so we can get a lot of feedback, and the video off. It just makes it a lot easier. And because we have quite a large turnout, if you have a question during the webinar, just type it into the chat area, and I'm going to read those off to Glenna when she gets to the ask a question section. So just feel free to type in a question during the webinar, and I'll have those all collated for Glenna when we get to the question section. And you can type in as well when we get to those sections. This webinar is brought to you by the BC Goat Association, and we just like to point out a few things that benefits of being a member of the BC Goat Association. We have government representation at the provincial and federal level. We provide a newsletter to all our BC Goat Association members. Obviously, we provide webinars and a lot of education as well. So we'd really like to welcome Dr. Glenna McGregor. She works for the BC Animal Health Centre, and she's one of the top veterinary pathologists here in BC. She also works as a professor at the Western College of Veterinary Medicine. And I have to say, she's extremely passionate about educating everyone on small ruminants. And I really appreciate her passion for that education, because it's a huge benefit for producers and for animal welfare. So having said that, I will pass it over to Glenna.
All right, thanks, Zoe. Technology is not my forte. Let me try and share my screen. And present the slideshow. There we go. All right, hopefully everyone can see that.
Yes, I
It's perfect. Awesome. Yeah, thanks so much to Zoe for organizing this. It's been something, you know, it's one of those things I keep thinking, oh, we should do a webinar while everyone's stuck at home, and then it never happens. So many thanks to Zoe. And also, yeah, just echoing her plug, joining your local goat association if you have goats, or the sheep federation if you have sheep. Honestly, it is like the best value for money you can have. Their representation in government that's who we reach out to if there's a problem that's affecting sheep or goats. We reach out to Zoe and then to Barb with the Sheep Federation, or if there's legislation involving them, and they do a ton of work consulting on that sort of thing. The education is phenomenal, and it's just a really lovely group of people. So I'd very strongly recommend joining your local association and federation. Yeah, for everybody, even if you don't have sheep or goats yet. It's just the magazines are great. Just it's really great being plugged into that world. If you're at all interested in sheep and goats, and if you aren't interested in sheep and goats, you might want to go watch Netflix or something, because you probably wouldn't be here. So I'm a pathologist, veterinary pathologist at the Animal Health Center here in Abbotsford. So for those of you who don't know, it seems like there are quite a few people who are maybe a bit new to agriculture. The Animal Health Center is on the east side of Abbotsford. So Chilliwack's over here. So right as you come into Abbotsford on Highway 1, you've probably seen it, sort of the very odd looking barn-like red building, just on the north side of the highway as you come into Abbotsford there. So that's the Animal Health Center. Probably about half the building is composed of the lab, which is where I work, and then the other half is government offices. So at the Animal Health Center, what we do is we diagnose disease and contribute to the management of disease and decreasing the risk of disease for animals in the province, and then also by extension, impacts on human health as well. So we've got, I didn't count, we have 9 pathologists. So every pathologist is a veterinarian who has then gone on for at least an additional 3 years of postgraduate education in the study of disease. So that's primarily, whoops, I did not know that was on auto switching. So primarily that is we do post-mortem examinations, figuring out why an animal died, and then how do we use that information to contribute to animal and human health in the province. So with sheep and goats, the pathologist you'll be dealing with will be one of these 4. Anne Britton, she's one of the leading experts on equine pathology. We all do all species, well, all species of mammal. Chelsea Hemsworth, she's also a professor at UBC, does a ton of work with rat surveillance. Excellent pathologist. Stephen Raverty, worldwide expert on marine mammal pathology. There he is wading in a whale. One time he came into my office and he was like, oh, Glenna, can you cover duty? Finland called. They have a whale that they want post-mortem. He's known around the world for that. And then that's me in the corner. We have 2 avian pathologists. So any of you who have small flock or large flock, poultry will probably work with either Vicky or Tony. And then we have 3 fish pathologists who deal with our aquaculture industry in the province as well. So that's Gary, Fanny, and Michael. Every now and again, Michael or Fanny will cover some regular duties, so you might see them on your reports as well. So contact information, honestly, the easiest way to find us is just Google BC Vet Lab, and it's the first thing that will pop up. We do have a website, which honestly is kind of an embarrassment. It's a bit challenging to navigate, but everything is there. There's a submission guide, which gives you a lot of really good information. And then there's submission forms in the corner. Any questions, just email this email or phone this phone. We are here from 8:30 to 4:30, Monday to Friday. And there's someone answering the phone all the time. When you phone, if you're have a question for a veterinarian, is typically not instantaneous, but it's typically same day. And if it's an urgent thing, we'll really try. If it's less urgent, it'll be same day, next day, if you have a question for one of the veterinarians. So the Animal Health Center services, what I'm most involved with is post-mortem examinations. So that's basically autopsies on animals, and then giving back that information on why that animal died and what else is going on with it to help figure out plans for other in-contact animals. We've got a parasitology section, and I'll talk about each of these in more detail. Serology, which is more of our herd screening, which we'll talk about it in quite a lot of detail later on today. Bacteriology, molecular diagnostics, so that's using DNA, PCR-based tests to diagnose different infectious agents. Virology, we've got an electron microscope. We do consultations all the time. And then before COVID, we had a series of keeping flocks healthy workshops. So I ran the sheep and goat one. Tony and Vicky did one on small flock poultry, sort of traveling all around the province. Paul Van Wessendorp did one on bees. And then we were in the midst of developing one on small herd pork production. That they are talking about putting that in an online version, but I'm not really sure where that's at. If you do want to send in a sample, we accept samples from veterinarians as well as from the general public. Basically, anyone can submit a sample who's from BC. The sample needs to be well labeled and have a completed submission form, and the submission forms are on our website. To get stuff here, depending where you live, direct delivery is often the best option. Here's a whale that was direct delivered. We are open 8:30 to 4:30 Monday to Friday. Drop off a sample anytime. If you don't have the submission form, we can guide you through filling the submission form when you get here. So if you live close, great option. If you don't live close, you are somewhat reliant on the mail, which is hit or miss at the moment. With increased shipping amounts, we're getting some delays. But Canada Post, Purolator, FedEx, all deliver to us. Ideally, if you're sending something by mail, ship early in the week, because the mail depots aren't refrigerated and things can rot really quickly over a weekend if they don't get here before the end of the week, because we aren't open Saturdays. If you are shipping something in the mail, it cannot leak or they get very, very upset, and often will discard the sample. So double bag, tape the lids of jars, that kind of thing. It cannot leak in the mail or it won't get here. So pathology is the study of disease. And the reason I think it's important is that you can learn so much about animal health, animal disease from studying the animals that have died. And that information can be used to apply to that animal's flock health, that animal's herd health, and it also applies to our provincial animal health and as well to human health. So we'll give some examples of that. Here's Tony. We get vet students from all over the world who come spend 2-week rotations with us. So this is Tony teaching a vet student. I believe he was from Amsterdam. Before COVID and before we all were wearing masks. Whoops, too much, sorry. So these are some of the sheep and goats cases have had in the last 2 weeks, just as sort of a smattering of the things we're diagnosing in sheep and goats. By no means representative of what we see commonly, just what I happen to have in the last couple of weeks. And just to illustrate why sending an animal in for a post-mortem exam might be helpful to you as an animal owner. And then it's also helpful to us as a province. We'll go through pricing in a little bit, but it is highly taxpayer subsidized because the information we get from the surveillance of the animals that people are submitting because they've died is just invaluable to us as a province in terms of managing animal disease. So up right here, I don't know if you can actually see my cursor. Up right here is a goat that was submitted, sudden death. Here's the stomach of the goat. Oh, I should say there are some graphic photos. This is a bit late, but if you're squeamish, close your eyes. And then here are a ton of worms. So these are the barber pole worm. So this goat died of hemoncosis. So this is going to be affecting the rest of the herd. When you've got a worm burden this high, it's going to be affecting the rest of the goat herd. And it is going to be quite easily treated with a deworming medication. So that's really good information for the farmer to know. This goat did happen to be on a regular deworming protocol, but we've got really high levels of resistance to different dewormers. So they're going to have to switch to a different dewormer. And they're going to have to be really on top of making sure to test their animal after they've dewormed to make sure that their dewormer is doing what they think it's doing. Top left here. So this is the intestine of a goat, also presented for sudden death. And I don't know how many of you have looked at intestines of your goat, but they really don't typically look like this. They're usually a lot thinner and the wall is really smooth and shiny. So this is the inside of the intestine. You can see it's really rough and it's got some yellow stuff stuck to it. So this is Yersinia pseudotuberculosis, so enteric yersiniosis, which we don't see very often, but it is also really important for this producer to know because it can cause pretty serious disease in people. So they're going to really want to step up their hand washing and their hygiene around these goats to make sure that they aren't infected as well with the Yersinia pseudotuberculosis. Also, they're going to want to watch their other animals because this is going to be treatable by an antibiotic, but only if you get it in time. Bottom left here, that's sort of that's a sheep. Sorry, this was for the Goat Association, so most of my examples are goats, but I put a sheep in for you. So this is a head of a sheep that's been cut sort of right down the middle. So nose is up here. This is the brain right here. Neck would be down here. And right here, you can see there is a giant mass. So what this is an enzootic nasal tumor. So it's a tumor that is triggered by a virus in sheep. So this is something that's going to be pretty important for the producer to know if she's trying to sell her animals, knowing that they've been exposed. And if she does have some to start developing clinical signs, she'll have a better idea of what's going on. And also this is pretty uncommon in sheep in BC. So it is useful as a province to know that we are seeing some of it. You know, and if we start seeing an increased incidence, that's something where we might reach out to the BC Sheep Federation and say, hey, we're seeing increased enzootic nasal tumor. Maybe we should do an article in the Sheep Federation magazine or something like that.
Glenna, I'm just going to interject for a second. Somebody was asking, they're not sure what the pseudotuberculosis that was. If you could just tell us what it is in layperson terms to know what type of disease it is.
It's called enteric ursiniosis. There isn't a lay term because it's not really that common.
Okay.
Yeah, there's really no lay term. But I will try for the other ones if there is a lay term for sure.
Okay, perfect.
So then this one on the right, sorry, I didn't have a picture of the uterus. This is a microscopic picture of the uterus, and all the red is bacteria and inflammatory cells. So this was actually a really interesting case. This goat aborted 3 fetuses, triplets, but then just didn't come right and eventually died. And on postmortem exam, there was a really severe uterine infection. We did bacterial culture of the uterus, and we came up with a bacteria called Truparella abortosuus, which is actually really, really interesting because it's never been reported in goats before. So this is something where producer got some good information about what killed her goat, maybe not as much as she would have wanted because we don't know very much about this pathogen. It hasn't been reported in goats before. We can't really tell her what to do with it, but we're also going to publish it, and that's going to be added to our general knowledge of humanity's knowledge of things that can cause abortion in goats. And so, you know, once that's published, other people will feedback to us from other parts of Canada and other countries will be like, hey, we saw this too. And we can sort of start to get a better idea about what this bacteria does, how common is it, does it often cause abortion in sheep and goats, and sort of contribute to like sort of the greater veterinary medical knowledge. So there's just some cases. We do several sheep or goats every week. And so that's just from the last couple of weeks. As a lab, this is just sort of why I think my job is cool slide. Not a lot to do with sheep and goats, but we do a lot of other things as a lab. So this on the top left, we do quite a lot of wildlife surveillance. So here I'm testing a bat for white nose syndrome, which is a disease that's threatening bats. We don't have it in BC yet, but it has caused massive bat die-offs throughout a lot of North America. In the middle here, this is those of you who live on the Gulf Islands may have heard of the white and black-tailed deer die-offs that they were having, I believe, on Maine and Salt Spring Island, caused by deer hemorrhagic disease, which was diagnosed for the first time in British Columbia this spring. So here's the deer hemorrhagic disease, and this is sort of the smudgy purple here in the nuclei of this vessel. Those are viral particles. So new incursion of a disease that's been known elsewhere, but never known in BC before. Up here, we have a picture of, this was a parrot-like bird that was brought in from overseas and was being quarantined and died. And I did a postmortem, and again, you can see this purple smudge are viral inclusions within the liver of this bird. So this is adenovirus. This is a disease we don't have in Canada. So catching it before this bird left quarantine was pretty nice. Up here, this was part of a homicide investigation where we had some dogs and some people found dead in a burned out structure. And the question was, were they killed and then the structure burned, or did they die in the fire? And the humans were too decomposed or too badly burnt for the human pathologist to tell that. So they were hoping the dogs, it was the same thing. And here you can see there's soot tracking down the trachea, meaning that they did breathe during the fire and died during the fire. We're also involved in animal cruelty investigations. So if an animal dies and cruelty or neglect is suspected, it will come here for postmortem exam, and then we'll, if it does proceed to charges, we will testify. And then one of our main roles is sort of a sentinel for emerging animal diseases. So African swine fever is one we're really worried about. So we had this picture of a pig, which this looks like African swine fever. It wound up not being, but it was really important to get this pig into the lab and see if African swine fever had come to Canada, because that would definitely close our borders for pigs. So sentinel for reportable diseases and diseases that are present outside our borders and we don't want to have inside our borders is a major part of what we do as well. Sort of alluded to the role. I think with COVID, we're all becoming more and more familiar that diseases in animals don't stay in animals, they can move to people. COVID did not originate in people, it originated in some sort of animal host that's yet to be determined. And so I think there's more knowledge now about diseases that are shared between animal and people. But that's another part of what we do here is try and manage human disease by surveying for it in animals. So top left, this is a bat brain. This here is a bat neuron, the gray, that's sort of the purple thing. And then in the purple thing, you can see a pink thing. That is a rabies virus inclusion. So this bat has rabies. Down here, this is a crow with West Nile virus. In the spring, all summer really, the most sensitive indicator of when West Nile virus has entered into a new area is going to be dead crows. So we do quite a lot of dead crow surveillance looking for West Nile virus, because then if we find it, we can tell people to please, put on bug spray. There is West Nile virus in your area. This was, I believe, from the Kootenays. Middle here, this is salmonella. This is the liver. You can see all the little white spots on it. This is the gallbladder. It's got some yellow gelatinous stuff on it. Very typical of salmonella. This is in a dairy calf. Up here, Cryptococcus gattii in a porpoise. This is the goat one. So many of you may have heard of Coxiella burnetii or Q fever, which causes abortion in goats. We don't see it all that commonly, but it certainly is around. This is an important one to diagnose because it can cause fatal disease in pregnant women. It causes flu-like disease in everyone else, but in pregnant women, it can be very dangerous to the fetus. Mink are something that have occupied a great deal of my time for the last 3 months since COVID was diagnosed on a mink farm in the Fraser Valley. So we've got a pretty big COVID surveillance program in mink. So I spent a lot of time swabbing mink noses recently. So for necropsy, if you do have an animal that you want to submit for necropsy, of course, it's entirely up to you if you want to submit it. You're always welcome to give the lab a call and say, this is the scenario, this is what's going on. We are not paid by what we bring in. So I have no incentive to give you bad advice and tell you to bring in an animal if I don't think it's going to be helpful. So if you're not sure if it's worthwhile sending something in, give me a call and be like, okay, this is, check this. So it looks really pale. Okay, just send in a fecal, look for worms. You don't need to send in a whole animal. Or like, yeah, that's something that sounds like a necropsy could be really helpful, and we could come up with a diagnosis that would really change your management. So if you're unsure whether you should send something in or if it's going to be worthwhile, give us a call. We have no incentive to lie to you. We have no incentive to bring more animals in, whether I work or not. I'm paid the same. So cost-wise, you're looking up to for $140 for up to 3 animals with the same disease process. So 3 animals that are around the same age, same clinical signs, and owned by the same person or kept in the same location. So most commonly, this would be for lambs. Say you lose 3 lambs in a season. If you send all 3 of them, we're going to get a better picture of herd health than if you had just sent 1 in. If for companion animals, so pets, horses, it is $250, both of which are honestly a steal of a deal, because these include, no other lab in Canada does this, but it includes up to 5 tests that are needed to determine the cause of death at the pathologist's discretion, which is actually so nice as a pathologist because I get to go further on the cases and I get to just make sure that I'm right. So instead of being like, well, it looks like rotavirus, I can test for rotavirus and be like, looks like rotavirus, it is rotavirus. So it really just, it keeps me honest with my diagnoses. And so in terms of cost, depending on how complex the case is, you're probably looking at $500 to $1,000 of value with every postmortem you send in. We're heavily taxpayer subsidized. So it's really good value for money. Though I know profit margins are thin, and each farm is going to have to come up with their own discretion when it's worthwhile, when it isn't worthwhile. Times for sure you want to think about sending it in if you're losing multiple animals and you don't know why. If you're new, this is going to be how you learn. You learn, in death we learn. So you are going to learn from the animals that you lose. You're going to learn a lot about mineral status, worm status. If you're new, it is good to be err on the side of sending more in. So histology, this is what I do pretty much for every case. So every case that comes in for a postmortem exam, I will examine with my eyes. I'll look at every organ system, what I can see with my eyes, and I'll take samples of each tissue and look at it microscopically. And that's really helpful for figuring out if an infectious agent is causing disease. So this is caprine arthritis and encephalitis virus, or CAE, in a goat. So bottom right here is a normal lung. This is a lung of a goat that has clinical CAE. So a lot of goats who have CAE won't have disease. If it causes a disease, it looks like this in the lung. So you can see normally the lining of the alveoli, so the little air sacs in the lungs, are extremely thin, so oxygen can diffuse across it. With CAE, you get these really plump cells that oxygen just can't get across a cell that's that thick. So, kind of already went through this. So consider submitting for necropsy anytime you have an animal that dies and you don't know why, particularly if you have multiple animals dying or you're worried about a herd problem or flock problem if you have sheep. I see a question, can I just send in a liver for mineral status? We don't do mineral analysis here. Unfortunately, our toxicology lab was closed down for fiscal reasons, but you can send directly to PDS in Saskatoon for mineral status. You can send it to us for mineral status, but then we'll just charge you shipping and handling. So it's easier if you're just wanting to do mineral status, send directly to Prairie Diagnostic Services or PDS in Saskatoon. The animal does not need to be frozen before sending. It really depends where you live. So if you can get it to us within about 24 to 48 hours of death, keep it fresh. Freezing does destroy the cells in a way that makes the microscopic examination a bit more tricky. But also rotting does destroy cells in a way that makes microscopic examinations tricky. And right around that 48-hour mark is where we get, being frozen is less hard to read through than being rotten. So if you can get it to us within 48 hours, send it fresh. If it's going to be more than 48 hours, the animal dies as it does on a Friday afternoon, then freeze it and send it in. Also, if you're coming from a long way and you're shipping where it's going to be in transit for quite a long time, also nice to have it frozen so it can sort of thaw in transit instead of being warm and rotting in transit. If you have, we get this question all the time because there's some nuance to it. So if you aren't sure whether to freeze or not before you send, give us a call and we can give you advice on that. In general, if you can submit the whole carcass, that's often better because we do this all the time. We are going to be better than your private practitioner, and we're going to be better than you are at detecting abnormalities. Just because we do it all the time and we see it all the time. So if you can send a whole carcass, that's great. If you're in the Kootenays, sometimes shipping and you have a large animal, shipping is going to be cost prohibitive. You don't want to do a road trip to Abbotsford. So then you're going to look at sending in a necropsy in a jar. So collecting samples and sending small samples of tissues into us. Typically, it's better if you can get a veterinarian to perform the postmortem and pick the small pieces of tissue, especially if you aren't familiar with anatomy and aren't comfortable with that process. If you hunt a lot and are familiar with anatomy and are comfortable with it, give us a call. We have a protocol for how to do a necropsy on your own, and we can email that to you. Unfortunately, it was deemed too graphic to be posted on the internet, so we can email you that protocol and you can work your way through it. We often get a diagnosis when people do the postmortem themselves, especially if they really follow this protocol and take the photos they're supposed to. We don't get a diagnosis as often as we do with an animal that's sent in as a whole carcass, but for practicality, sometimes it just isn't an option to send in the whole carcass. So in terms of abortion, I know Zoe was saying that was a question some people have questions about, especially in sheep and goats. Figuring out why your animals are losing fetuses or having an abortion is fairly important because there are quite a few infectious agents in sheep and goats that can cause disease in people, including 2 that cause fairly life-threatening disease in pregnant women and can result in fetal death. So more so than other species, figuring out why a sheep or goat is aborting is important. Abortion diagnostics are frustrating. So only about 30% will you get a diagnosis. If you can submit placenta, there's some disease conditions that really only affect the placenta. Typically, we accept about a 1% rate of abortions as normal. So as soon as you're losing more than 1% of your lambs or kids, that will be when to submit. If you're not sure how many you're going to lose, put them in a freezer. Say at the end of kidding season, lambing season, you've only lost 1%, then you can toss it. But just keep each one because, you know, okay, you lose one today, 3 days later you lose another one. It's going to be really helpful to send those 2 animals in rather than just one to get a more representative look at what's going on. This is a very long list of the causes of abortion in sheep and goats. Everything that is written in red can cause disease in humans. So just to emphasize that it is pretty important to figure out why a sheep or goat is aborting. We basically can figure out with the postmortem exam that we do, we can diagnose every cause of abortion on this list except maternal illness, which is far and away the most common cause of abortion, is that for whatever reason, the ewe or doe just aborts. Maybe they had a fever, maybe they were stressed. Could be, we don't know what's going on. Genetic illness is also something that we have trouble diagnosing. We are great at diagnosing anything infectious and fairly good at diagnosing any sort of nutritional deficiency or toxic issue. So when you get, when you send an animal for an abortion and you get the report abortion of unknown cause, which is 70% of the reports we send out for abortion of unknown cause, it can be really tempting to think that that was a waste of money and time, but we have ruled out basically everything on this list, and which are really important things to rule out. So parasitology, we get a lot of parasitology for sheep and goats. We have gone back and forth about how we're going to offer our sheep and goat parasitology because there's 2 things that are really important. One is coccidia. So upper right, we see an intestine of a kid that had coccidia. These little button-like knobs are proliferations of cells containing a single cell parasite, which is coccidia. So that causes, most commonly it causes decreased weight gains, just sort of poor doing. Occasionally it can cause diarrhea and death. So coccidia, we need to do a fecal float to look for coccidia. It's really hard to tell on a McMaster because you just aren't zoomed in enough for that. The other one is strongyles. So that's what causes, Haemonchus is a type of strongyle. It's sort of the umbrella classification of a bunch of different worms. Haemonchus or barber pole worm would be the most common strongyle we see, and the one that we worry about the most because it causes the most disease. So this here is a goat with bottle jaw caused by barber pole worm infestation. It is our most common cause of death in both sheep and goats that are submitted to us. And I feel like it's underrepresented because a lot of private vets and farmers are going to be able to diagnose this on their own, so they aren't going to send the animal in. So it is a significant cause of mortality in BC. So for strongyles, the fecal float is, I'll show you how we do them in a second, but the fecal float really concentrates the eggs. And so it doesn't really distinguish between a mild, moderate, and severe strongyle infection. And so for that, we need what's called the modified McMaster fecal egg count, where we come up with a count of the eggs per gram. So what we're doing now for sheep and goats, we call it the sheep and goat parasitology panel, where for the price of one, we do both of these. So we can catch the coccidia. We can also catch the eggs of different species that are just not as common. So things like capillaria or trichuris. And then we also provide you with a fecal egg count. We do this in alpacas and llamas sometimes, and I recently did one in a water buffalo. So we probably will be changing the name to something a little more inclusive, but it's sort of the same thing. We do get flack sometimes where the fecal float and the modified McMaster, the values may appear to differ, but they're really just measuring different things. So here's what we do with the modified McMaster. You take 2 grams of feces and then you put it in this which you mix up and turn into a liquid with a certain volume of fecal sol. And then we put it in this McMaster plate and then in the grids. And then you count how many eggs are in each grid line. So you use a set volume of feces, and then you count how many worms to get an estimate of the number of eggs per gram. The thing is, this is measured at much lower magnification, so it's really quite hard to see coccidia. So for our coccidia for the fecal float, we take not really a measured volume of feces, we dilute it down with Schiether's solution, and then we centrifuge it to really concentrate all the eggs. So you get a really good concentrate of the eggs. So the eggs that aren't present in as high numbers will show up, whereas they won't show up on a modified McMaster. And it's done at much higher magnification, so you can see the coccidia. So these are strongyle eggs, these are coccidia, strongyle egg, this is a trichurus egg. These are things that you can learn to do yourselves if you have a microscope and tolerance for playing with poop. It's actually quite easy, and if you have a large number of animals, that would be quite cost effective to do it yourself as opposed to sending it in. Sending it in here is $27 per sample. A lot of private vet clinics will also offer the modified McMaster, and definitely if you're looking at worm management, you absolutely need to do a modified McMaster. You need to quantify the number of strongyle eggs, because it just doesn't give you enough information with just a float. So small animal clinics typically will do just a float. Most large animal clinics will do the counts, which is what you really need to do. Magnification, so the modified McMaster, we do at 40 times magnification, so 4 on your objective lens, then we've got 10 on our oculars. This one we do at 100 time magnification. Doesn't have to be super high. So like just a school microscope is fine, like a high school microscope. It doesn't have to be a super good microscope. So times to consider parasitology, or at least checking your animals, and whether you do that by doing what's called a FAMCHA score, so looking to see how pale they are in their eyelids, or doing a fecal analysis. Use or dose 1 month prior to lambing or kidding. The key one is lambs at midsummer. So ideally you want to do about 10% of your lambs, or 10 animals, whichever is sort of the most, sort of mid-July, and then look at that every 4 weeks would be gold standard. That can be cost prohibitive. So if you're doing it yourself, do it every 4 weeks. If you're not doing it yourself, you might have to cut some corners. Definitely, we see so much dewormer resistance in BC, and almost all of the animals I see in that have died of barber bollworm have been dewormed at some point in the last few months. So definitely checking your fecal egg counts before and after deworming, and at least a couple of animals is really important to make sure that your dewormer is still working. There's other dewormers, there's going to be new dewormers on the market, but we are seeing a lot of resistance to the common dewormers. Breeding animals in the fall, if you've had trouble over the summer, new introductions are a big one because you don't want to bring in worms. And then any sheep that is thin or growing poorly, or has scours, especially if it's been on, when you first put them on pasture, everyone gets scours. They've been on pasture for a while and they've got scours. That's definitely a time you should do a fecal egg count just to check and see where you're at. So this is our bacteriology lab. It's a bit trickier in animals than in people because they do everything from saltwater fish, which have to be grown on saltwater media, a bunch of different species. So mostly what you're going to send in, as sheep and goat owners, would be abscess contents, and that would be looking for CL, so caseous lymphadenitis, which we'll talk about in a bit more detail later. Top right here, this is called a Kirby-Bauer plate. So when you send in a sample for bacterial culture, you'll get back a sensitivity result. What that means is they've taken the bacteria that grew from your sample and made this lawn with it. So the white background is a lawn of bacteria. And then they put these tiny discs on it that are impregnated with different types of antibiotic. And they see how far that antibiotic inhibits growth. So this bacteria would be resistant to whatever this little, sorry, I'm poking on the wrong screen, whatever this little dot is. Highly susceptible to, it's probably ciprofloxacin. This is ampicillin. This bacteria is resistant to ampicillin, sensitive to these. So when you get your sensitivity results, that is what inhibited bacterial growth on a plate in a lab. It doesn't tell you that antibiotic is going to work for your animal in a field situation, because different antibiotics penetrate different tissues differently. And so just because it works on a plate, sure, it might be susceptible to gentamicin, but gentamicin does a terrible job of swimming through abscesses, so it's not going to be effective in an abscess. You wouldn't really be using gentamicin in practice. So say tetracycline does a terrible job of swimming through pus, so it's not going to be effective for a CL abscess, even if it looks like the bacteria in that abscess is susceptible in the lab. So the sensitivity results are something you really need to interpret in consultation with your veterinarian when you're coming up with an antibiotic to treat your animals. So molecular diagnostics. So one of the most exciting advances in diagnostics in the last 30 years has been the widespread development of PCR tests. So polymerase chain reaction, all over the news right now with COVID. We have PCR tests for over 350 different pathogens. So we've got them for viruses, we've got them for parasites, we've got them for bacteria. And it's amazing because it can find very small pieces of genetic material of that pathogen, which, a lot of viruses won't grow in viral culture. And so you can detect stuff that there's no way you can detect really any other way. It's fast, it's efficient, it's really, really accurate. Interpretation-wise, it's so good at finding things that sometimes there can be a very small quantity of your infectious agent present, and it'll come up as a positive. And so it does take some interpreting when is a positive significant, and that's when it's really nice to pair it with the histology and be like, okay, I found this virus, and this sure looks like the pattern of viral injury microscopically. This is what's causing it. This is our molecular diagnostics labs. You've got two different types. So on the left is pictured what's called conventional PCR, which is sort of the old school way. It's easy to develop, cheap, and typically what we do with new pathogens. On the right, we've got what's called real-time PCR. It's way faster, it's hours, more expensive to run and more challenging to develop. So we do that for things where, like our COVID PCR is real time, our influenza PCR is real time, stuff that we need to diagnose very quickly and that we diagnose often because it's a lot faster, will be a real-time PCR. Not really relevant for when you're sending stuff in, but can explain some of the variation in our turnaround times for different PCR tests. Virology, we do very little virology in sheep and goats. We basically only do virology when we don't know what's going on. So we've got 7 different cell lines that we keep alive in the lab. So we've got ones from cattle, sheep, goats, chickens, horses, canine, and then a couple ones for weird animals. So different cell lines, and then you'll inoculate your tissue onto that and see if the virus is killing those cells. We also do egg inoculation and have an electron microscope. These are tools you would not use. Maybe if you send in an necropsy, something really, really weird, I would proceed to viral culture electron microscopy. But basically, this is when you're looking for something new, so some sort of new infectious agent that we don't have established diagnostic procedures for. Serology labs. Serology is where you're going to do a lot of your herd screening. So what that is you're looking for antibodies. So you get these multi-well plates, and so they're coated with, say, what's a common one? Johne's disease is a common one, and it's an ELISA. So it's coated with the Johne's disease bacteria, and then you put the blood of the animal on, and if that blood contains antibodies, it will bind to the Johne's disease bacteria that's coating this well, and then you'll add another antibody that targets that antibody that's stained blue. So the bluer this stain, the more antibodies are present in that animal's blood. And that'll tell you whether an animal has been exposed or not exposed. So serology is looking for antibodies, so it's looking for immune response. In some diseases, like Johne's disease and CAE, which are lifelong infections, if they have an antibody response, 99.9% of the time they are actively infected with the bacteria in the case of Johne's disease or the virus in the case of CAE. Other things, they could have cleared it and still have antibodies for it. So CL, sometimes that happens, so caseous lymphadenitis, sometimes they'll have antibodies for it, but they will have cleared the infection. And so you'll get a positive in an animal that's negative. Also tricky with CL, if they're vaccinated for it, they'll have antibodies, and so they will test positive on serology. So serology is really nice. You can see this is, what is this? I think it's like a 96 well plate. I actually didn't count this one, but they're multi-well plates. And we've got these pipettes with multiple tips, so it's really fast to test a lot of animals. So for herd screening, it's fast, it's efficient, and it's cheap. So we'll talk about that a bit more detail when we're talking about surveillance plans for Johne's disease, CAE, and caseous lymphadenitis, or CL. Which brings us to CAE, so caprine arthritis and encephalitis, or Mycoplasma. So for those of you who haven't heard of this, this is a viral, they're two very closely related viruses. I'm going to talk about them together because they're very, very similar. Typically, CAE affects goats, Mycoplasma affects sheep. Sometimes there's a little bit of crossover, but both of these are very similar viruses. They're in the same family of viruses as the HIV virus in humans. So they're in the retrovirus family. They do not cause disease in humans. They're just diseases of sheep and goats. But they proceed very similarly to HIV/AIDS, where an animal will be infected and it can take years before they show clinical signs. Sometimes they'll never show clinical signs. If they're going to show clinical signs, there can be a real range. Often you'll see, so the lung histology I showed, I think that was CAE. It might have been Mycoplasma, they're very similar. You'll get a pneumonia, but a pneumonia where there's not phlegm in their throat, so you don't sort of get that coughing that you would if there was phlegm in your trachea, like you would from a bacterial pneumonia, but just the lung is not working oxygen just is not diffusing across that lung. So they'll be really struggling to breathe, but you probably won't see coughing. And then you'll often see an animal that's very thin. Particularly in goats, you get what's called hard bag. So their udders just get really hard and firm. You can get arthritis. That's very common in older goats with CAE. You'll get an arthritis, especially on their knees, so the wrist joints. Particularly in younger animals, you'll see neurologic signs. So they might, like this one sort of got a kinky neck, and this one's head pressing. So you'll see some neurologic signs with CAE. And we definitely see it, but we don't see a ton of CAE in BC. Much less, I used to work in Ontario and we saw it a lot more there, but we still do see it. So if you are bringing in animals, I would suggest testing for CAE. Obviously up to you, of course. So we've got two tests, PCR. PCR is pretty much, we only use it on dead animals because you need to have the affected tissue. So you're not going to use that for herd surveillance. Luckily, we have an excellent serology test. So it's an ELISA. The thing about CAE is once you're infected, you don't clear it. You're infected for life. So if you have an antibody to CAE, you have the virus. So according to the manufacturer, there's about every 1,000 positive results, 4 of those will be false positives, which is actually really good. So it's really rare to get a false positive. The exception is in young animals because antibodies pass in the colostrum, the first milk. So a young animal will often have some antibodies to CAE if the doe or ewe is positive, and they may not be infected. So less than 6 months of age, the test's not very reliable. It has good sensitivity, especially if you're doing a bunch of animals in a herd to try and say, yes, this herd is negative. Occasionally in the first few months of infection, before the antibodies have had a chance to be developed, you might see some false negatives. So the suggestion is to retest twice a year for the first year and then annually thereafter if you want to be really, really confident in saying that your herd or flock doesn't have CAE. Cost-wise, I think it's $7.50 a sample. So I have it right here. Oh no, it's written that price varies with test. Somewhere between $7.50 and $10 per test. And there's a discount for bulk, but I can't remember what it is. It's on our website. Johne's disease. This is actually really common in BC. It causes, in cattle, you may, if any of you have cattle, you definitely be familiar with Johne's disease. It's very common. It causes a hose pipe diarrhea and wasting. Goats evolved as desert animals, so they are extraordinarily efficient at absorbing water from their intestinal tract. So you often won't see the diarrhea in a goat. You might see some clumping. So an absence of diarrhea in a goat doesn't mean that Johne's disease isn't there. But you'll see these really thin animals, especially a really thin animal that has a good appetite. So this here is the intestine of a goat with Johne's disease. And all this sort of, so these are the villi that line the intestine and are responsible for absorbing nutrients from the intestine. And then here's a bunch of inflammation. And so that's it, they're just not able to absorb nutrients. So that's why you see an animal with a fabulous appetite and skinny, skinny, skinny. And here are the little purple things. Those are all the bacteria. There's just tons of bacteria typically in a goat's intestines with Johne's disease. Oh, I see a question. How can we package red top tubes for CAE if we do it ourselves? Ideally, you need to spin them down and take the serum off. And the reason for that is because if the red blood cells in the blood break, they'll stain the serum red. And as you can see, our serology test is based on how blue something is. And so if there's a red color in from broken red blood cells, it really messes in our ability, because we use a spectrophotometer to tell how blue something is, and it just screws it right up. So you'll need to let them sit so the clot forms, so sit for like 10 minutes, spin in a centrifuge, take the serum off. If you can get it here within 24, 48 hours, just an ice pack is fine. If it's going to take longer, or if you're sort of having a prolonged collection period, freezing it is just fine as well, and then sending it in frozen. And it can be kept frozen for at least a year without degrading. So for Johne's disease testing is a nightmare. And it's really complicated, and there's quite a lot of nuance that's a bit case specific, which of the two admittedly fairly bad tests is going to be the most effective. So it is one where it is worth either phoning us or talking to your veterinarian, maybe both, to figure out which of these two tests is going to be, or if you're going to want to do both of them, which of these tests is going to be most effective for what you're trying to achieve with Johne's disease testing. So fecal PCR, we can pool the feces of up to 5 animals under a single charge. So a charge is $35, that's $7 an animal. So and our fecal PCR tends to be a little more accurate than the blood test. Both of them are actually pretty terrible. Both have a lot of false negatives. But typically, if you want to prove that a herd or flock is free of Johne's disease, doing fecal PCR pools is going to be your most cost effective and your most accurate and likely to pick it up if it is present. Very rare for there to be a false positive. So 3 in 1,000 positives, or if you have 1,000 positive, 3 of those will be false positives. Very uncommon. Sensitivity, so the number of false negatives, they are very, very common, and it depends on your stage of disease, because what the fecal PCR does is it picks up the bacteria that's being shedded in the feces. And early in the course of infection, there's very low levels of bacteria being shed in the feces. So late stage infection, so if you have an animal that's very thin, it's 76% sensitive. So you'll have, what's that, a 24% of your negatives will be false negatives. So still not amazing, but okay. If you're looking at an animal that's young and they tend to be affected very young in life, but then it doesn't grow to be a problem until later in life. So if you're testing an animal that's very young, it's got a 4% sensitivity. That means 96% of your negatives could be false negatives. So in a young animal, honestly, is not even worth testing. Serology, ELISA, on an individual animal level is cheaper. Again, also very specific, so false positives are very uncommon. It tends to be a little less sensitive, and these could be differently sensitive too. So if you pick it up, sometimes you pick it up on fecal PCR, sometimes you'll pick it up on the blood test, the serology. So sometimes if you really care about a single animal, doing both, it can be really helpful because it just increases your chance of picking it up. Again, it really depends on your stage of disease. So in an animal that is in quite advanced disease, fecal PCR is going to be your choice. If it's an animal that is quite early and affected, serology is going to be your choice. But again, it gets really complicated. So if you have questions about it, or you're not sure which test is going to be best for you, it's best to call the lab, talk to one of us, and we can guide you through that decision sort of on a case-by-case basis, or talk it over with your veterinarian. Typically, if you're doing a flock eradication program where you want to test and see which animals in your flock or herd are infected, typically it's dairy goats that do this. Serology is going to be the most effective and cost-effective way to do that, at least on an initial screen, and then maybe drilling down into fecal PCR, sort of when you're trying to prove that you've gotten rid of it. But again, check with one of us. It's really quite complicated because neither of these tests are very good. It's a really difficult disease to detect early. So again, complex, many nuances. Talk to your veterinarian or call us and talk to us before you come up with a plan, just because it is complicated. Both tests are going to be more sensitive in animals that have advanced disease. So if you want to screen your herd and you want to do it in a cost-effective way, test the oldest and thinnest animals. So ideally at least 10 animals, which is really only 2 pools of feces, or 10% of your herd, whichever is greater. Ideally, if they've mixed, will give you the best idea. Don't even bother testing an animal that's less than 14 months of age. The test just isn't good in animals that young. Even if they're positive, it'll test negative. So don't even bother testing. Caseous lymphadenitis, so or CL, more commonly known as CL because it's a bit of a mouthful, is caused by the bacteria Corynebacterium pseudotuberculosis, different from the ursiniosis, same long obnoxious last name, but different bacteria. It causes abscesses. In sheep, it tends to be spread by shearing, so the abscesses are often under the skin, often in areas of lymph nodes. In goats, they often get, because goats eat everything, they always have cuts in their mouths. It tends to enter through cuts in their mouths, and so you get the abscesses tend to be much more distributed throughout the body cavity of the goat, and you won't see them on the outside. So here's, so heads up here, oh, I'm not on the wrong side. Heads up here, tails down here. Here's the liver, just with a ton of abscesses all through it. And the abscesses have a fairly characteristic, it's kind of like ricotta pus, often has these sort of layers to it. It's a very, very cheesy pus compared to a lot of the more liquid pus. For testing, if you have an abscess and it is oozing pus, culturing that is going to be by far your best option. If you post on the Facebook, which never gives you bad advice ever, that you have a sheep with an abscess, guaranteed someone will say you just have to cull it. That is not true. About 50% of the abscesses that we culture from sheep and goats with suspect CL do not have CL. They have some other bacterial abscess that is super easy to treat. So don't cull an animal until you've cultured the abscess and proven that it is CL. CL is extremely hard to treat, and depending on your management style and what your goals are, you might look at culling a positive animal, but that's a conversation for you to have with your veterinarian, or with us if you don't have a veterinarian. But it's going to be very management and person specific. Yes, I'm getting on to the serology for the blood test right now. So serology, unfortunately, there is no, so serology, again, that's testing for antibodies. There is no serologic test available in Canada at the moment. There were, earlier this year, there were 2 commercial test kits on the market, which we were really excited about, and we were just about to bring them in, but then they were pulled from the market because they weren't very good. So right now, there's only 2 labs in North America able to do CL serology. One is, that should say UC Davis, University of California, Davis in California. And one is at the Washington Animal Disease Diagnostic Laboratory in Pullman, Washington. So we send to UC Davis. You are free to send yourself to UC Davis, but honestly, shipping biologic specimens across the border is a bit of a nightmare. We have a dude that does it, that helps them get across. So it probably is, if you're doing mineral panel, shipping direct to Saskatoon is going to be your cheapest option. It's going to be easiest if you send serum to us, and we will deal with the shipping across the border. Unfortunately, shipping is the most expensive part of our CL testing costs. So anything you can do to do a bunch of animals at once, join up with friends. We do try and batch ship them, but depending how many tests come in, we can only wait so long because you need results. So CL serology is good for herd health screening. There are about 1 in 20 positives will be a false positive. So if you're trying to test an individual animal, it's really quite hard to interpret that result because, yeah, there's a 5% chance that positive could be a false positive. And there also are a significant number of false negatives. So there's too many false positives and false negatives to really be able to interpret the results on an individual animal. Where it really excels is for full herd or flock screening. Because then you have a bunch of animals, you're like, okay, I've got a 5% positive rate, that's probably a false positive. The main drawback with this test is that vaccinated animals will test positive. So if your animals have been vaccinated, or if you're buying in an animal that has been vaccinated, it will test positive. And the test can't distinguish a vaccine positive from a true positive. Is there a... Cows don't get CL. I mean, very rarely they do if they're like pricked directly by the bacterium, but very uncommon. Cows definitely get Johne's disease, but unless you've got, animals can't get infected with Johne's disease after a couple months of age when they aren't eating hay anyways. So unless you've got your very young kids and goats eating hay that a cow that's infected has pooped on it, there's virtually no risk in buying hay from a dairy farmer. There can be some risk if you're using dairy colostrum that hasn't been heat treated, but hay, no problem at all. Yeah, so that's CL. I think this might be the last one we're talking about. So Mycoplasma ovipneumoniae is a bit of a hot political topic in BC at the moment. So it's a bacteria that is carried in the nasal cavity and sinuses of sheep and goats, and the sheep and goats typically are perfectly healthy looking. You can't tell. What the problem is that if those sheep or goats mingle with wild sheep, it causes massive die-offs and pneumonia in wild sheep. So these are all wild sheep lungs with Mycoplasma ovipneumoniae. There can be some weight gain problems, some pneumonia in sheep or goats, but there's really not a lot of evidence in BC that it is a major problem. Our lung condemnations of lamb lungs in BC typically ranges from 0 to 3%, which is the lowest in Canada. And Ontario, it's 25% that they're getting their lungs condemned, mostly actually due to this bacteria. So I don't have a lot of evidence that it's causing a big problem in sheep or goats, domestic sheep or goats in BC. The real issue is that it can spread to wild sheep, and it is going to threaten our wild sheep populations. So for people who aren't in areas with wild sheep, you can just tune out for this section. But if you are going to be selling animals to people who are in areas where they do have risk of contact with wild sheep, or you do have sheep or goats in an area where there is a risk of contact with wild sheep, this is a disease to be aware of. So there are two options for testing for it. The one we do here in BC and the one that's best in sheep and goats, domestic sheep and goats, is PCR on a nasal swab. So take a nasal swab, just sort of up the nose with a Q-tip, and then we do PCR on it here. And that's a DNA-based test. This bacteria is super hard to culture. So that's why we do the PCR, so that looking for the genetic material of this bacteria. The tricky part is that a sheep or goat that's infected might not always shed bacteria in their nasal secretions. So they can test negative one day and then positive the next. So if you're doing a full flock or herd screening, if you've got enough animals, a positive means your whole herd or flock is infected. If you're doing enough animals, you'll have that sensitivity that a negative means something. So if you've got more than 10 animals that you've tested and they're all negative, there's a 95% chance that your flock is negative. If you've tested 20 animals, all 20 are negative, there's a 99% chance that your flock is negative. The trick comes when you're trying to tell individual animals, because they might not shed bacteria the day you're testing, and then 3 days later they're shedding bacteria. So if you're trying to test on an individual animal, for example, if you're trying to bring in if you have a negative flock or herd that you've proven to be negative, you've tested, and then you're bringing in a new animal, you're probably going to want to test that animal multiple times, 2 or 3 times, a couple weeks, a month apart, to try and increase the likelihood that you're going to pick up that bacteria on a day that it is being shed in the nasal secretions. There is a blood test. It's not recommended in domestic goats. It's just doesn't perform very well in domestic goats. And that's only available at the Washington Animal Disease Diagnostic Laboratory in Pullman, Washington. So for CL, how come head and neck lymph nodes are most commonly infected? And how often do you see it positive with hindquarter lymph node swellings? Yeah, head and neck is more commonly effective. I think, especially in goats, it tends to come in through the gums and the mouth, which have little cuts in them, and that lets bacteria in. And then it's your head and neck lymph nodes that drain your teeth. And so that's where the bacteria comes first, and that's where you see the abscesses develop. With sheep, it kind of depends where you nick them most commonly when you're shearing. Again, seems to be the head and neck. But if you are nicking them sort of the hind legs, anywhere where you'll see it in the lymph nodes that drains an area where you've gotten a nick and injected the bacteria into that nick. So sometimes we see it hindquarters. We don't often see the external form, the one where people have abscesses on the outside, and that's because people can diagnose that themselves. They don't send it in. We mostly see the internal form because all people are seeing is an animal that's not doing well and losing weight, and they want to know why because they can't see anything externally to explain it. So we most often see the internal form, but I think that's more of an artifact of what samples come into us versus an actual trend in BC. And same thing for the slaughterhouses. Like, people aren't going to send an animal to slaughter that has visible external abscesses. So again, they're also seeing the internal abscesses more often. So it's actually really hard to get a sense of the distribution of external abscesses with how we're collecting animal health data in BC, because we collect it through serology, through slaughterhouse surveillance, and through animals that come in through the lab. So I can't really comment on hindquarter versus front back external abscesses. Probably the people in the crowd who have more experience will know better than I will. All right, I think that's all I've got. So open the floor to questions. Either type them in or unmute yourself and ask a question, whatever works best for everyone. But I think we are also a little over the hour, so I'll turn it over to Zoe as well. Let me figure out how to stop sharing.
Yeah, so if anybody has a question, if you want to unmute yourself and quickly ask a question, that I think will work at the moment, because we're not having too many questions fly out from the chat yet.
I should say as well, like I gave my contact information on the beginning. Feel free to contact the lab, for the more specific questions. That's what we're here for. Contact us, email, phone, if you have questions, for sure. The best way to treat foot abscesses in sheep depends a bit on what's causing the abscess. What you really need to drain it though. So trimming down so it can drain, and then keeping it clean and bandaged is going to be your best bet.
Does anybody else have a question for Glenna? Oh, here we go. Somebody else.
Okay, super. Someone interested in a fecal with microscope? Yeah, that's something that's best done in person. That's going to be really hard to do virtually. But the Lower Mainland sheep producers, correct me if I'm wrong, Barb, I think they had a seminar. Smithers Farmers Institute did a seminar. So probably something that'd be easiest to work with your local group and just get either me or get just a private practitioner in to do it. It's just one of those things that you kind of have to be one-on-one to help.
Yeah, that was certainly something we were looking at with either the Vancouver Island Goat Association or the BC Goat Association, is putting one on when we're allowed to have more gatherings. There was a question earlier about dewormers. It said dewormers that you recommend for goats. That was from Suzuki.
A dewormer you recommend for goats?
Yes, that was a question.
It depends a bit on what you're resistant to, and that's kind of trial and error. Typically either an ivermectin or valbazin or one of the bendazoles. It grows. Oh yeah. Can you confidently diagnose some mineral deficiencies with skin coat abnormalities, or do you have to have mineral levels? The skin is an irritating organ because there's only like a few things it can do when it's upset. It can sort of get dry and flaky or red and itchy. So unfortunately, the skin just doesn't have specific enough patterns. So there's a few things that can cause all like dry and flaky. So you really do have to do mineral levels. There's really not a specific skin pattern for different. The one thing is a copper deficiency. Sometimes you'll see a white ring, but that's really hit or miss. Mineral levels are the way to go. on liver would be the way to go with your animals that you're slaughtering. Success with abortion vaccines in sheep. I actually don't know if anyone here is using abortion vaccines. We don't see, so the vaccine is predominantly for Chlamydophila abortus, and we don't see that much of it. Honestly, I wouldn't vaccinate my sheep. And then there's also a Campylobacter one, which we almost never see. So I wouldn't vaccinate my sheep unless I had a documented abortion by those infectious agents. Most of our abortions in sheep that we see, like 99.9%, would not be from something that the vaccine would protect against. So I wouldn't vaccinate unless you've had a documented Chlamydophila or Campylobacter outbreak. Might have missed it. What causes resistance and deworming with goats and sheep versus small animals? Oh, I think that's the $1 million question. I don't think anyone knows. Especially, I mean, dogs, cats, they're not kept in large flocks. They aren't, or herds, they aren't dewormed nearly as often. But why we aren't seeing it in cattle compared to sheep and goats, I have no idea. Goats are tricky because they metabolize drugs more quickly than sheep. So you have to give a higher dose, but then there's no labeled dewormers for goats. And so a lot of people are giving the sheep dose, which isn't quite adequate. So that might be why we see a bit more in goats, but maybe someone knows, but I don't know why it's so much more common. Is rotating common commercial wormers still recommended, or are they all not working anymore? There's a few schools of thought on this. There is a school of thought you should give. So there's sort of the two classes of commonly used commercial dewormers. So the ivermectin class, and then the white dewormers, so the albendazole, fenbendazole dewormers. Some people suggest giving both at once to try and minimize resistance that way. Other people say keep with the dewormer that's working until it stops working, and try and go a few years and then switch dewormers, and hopefully you get a few years out of that. And by then your worms will be sensitive to the first dewormer you use, and try and go a few years. There's sort of two schools of thought. The main thing is to do fecals after you deworm to make sure that they are working, especially if you're in Lower Mainland or Vancouver Island, where we see the most, and especially if you're seeing problems, and if you have, if you've got heaps of pasture area, you're going to be fine. But if you're in a small space, you have quite a few animals, that's when you're going to start seeing more problems. You can't rotate pastures effectively. Like Lower Mainland, no one can buy enough land to rotate pastures. So that's when you really have to be on it for monitoring effectiveness. Newborn lambs that don't receive colostrum from the mother, should you vaccinate to increase the immune system or not bother? They're not going to have an immune system that's developed enough to respond to a vaccine until they're about 8 weeks of age. So it really is just going to add more stress to a system that already is under stress by not having colostrum. Ideally, every lamb should get colostrum. So getting some frozen colostrum from your local dairy, or there's some commercial colostrum packages, like a Head Start, or there's a bunch of different ones. You should give them colostrum if they aren't getting colostrum. A lot of people deworm right after kidding. Do you recommend not treating due to resistance? Is there any time you do recommend routine deworming? So routine deworming, the one time, again, it's a tricky problem to solve. So there's a lot of opinions, and the more opinions there are, that means there's no right answer. So there's a few schools of thought. If you're having problems with worms, and if you had problems the season before, I would deworm either right around kidding time, because worms are creepy smart. They somehow know that the lamb is going to kid, or the sheep is going to kid. And so they produce a ton of eggs right at the time of kidding, which means that those eggs have developed to being an infective stage about 8 weeks, when the lamb is going to be most susceptible to high worm burdens. They're diabolical, really. So the one time where, if you're having problems, that just doing a routine dewormer that some people recommend would be to do right at kidding, to try and prevent that. They call it the periparturient, so the right around time of birth egg rise would be the one time of year to do routine deworming. Again, probably if year to year, you're going to know. So if you had problems last year, then yeah, I probably would do it. If you've never had problems with worms, if you're on a pretty extensive system, you're not Lower Mainland or Vancouver Island, you're somewhere where there's less worm problems, I wouldn't. But if you're in a small area, can't rotate your pasture really well, you had some problems last year, I probably would deworm right around the time of kidding or lambing. Is there a way of testing blood for mineral deficiency as a preventative? It's tricky. It depends a bit on the mineral. So lead is super easy to find in blood. Selenium, not so much. Copper, copper levels in blood. So your body will really regulate the copper level in your blood, and the liver takes up the extra and has the deficiency. So your liver will be completely devoid of copper before you have any hint that you're low in copper in the blood test. And same thing with the excess. Like once excess copper is spilling over into your bloodstream, you basically have a dead animal. So copper, you can't tell on. You need a liver biopsy or dead animal liver to test for copper, unfortunately. You really can't go wrong with a commercial dewormer. I mean, you can go wrong, but if you don't know a lot about sheep or goats, going with commercial dewormer, because they are balanced, and they typically are balanced based on salt content. So animals will eat, just like us, you'll crave salt, and you'll eat sort of to a certain amount of salt, and animals are the same. So they balance the mineral based off of salt. So providing a commercial mineral that's balanced and then not providing extra salt, because then they'll satisfy their salt craving on the non-mineral salt and your dose will be off, is usually the easiest way to go. So you can't actually enter a dose with, and copper's tricky because it's such a narrow range between adequate and fatal. Especially in sheep, definitely use a sheep mineral. Goats, we rarely see copper toxicity. So I just use a goat or a cattle mineral is going to be fine. All right. So we are 15 minutes sort of over the hour. Maybe wrap it up there, Zoe?
Yes, well, thank you very much, Glenna. I really appreciate you taking the time to answer everybody's questions and taking the time to do this presentation. It's been a fantastic learning experience. And I've learned a lot as well during this webinar, so I really appreciate it. So thank you again for taking your time to talk.
Thanks everyone for coming and braving Zoom fatigue.
Yes. All right, and thank you for everybody taking this opportunity to learn more about herd, about your herd health and about the diagnostic lab, and it's great for improving your production and also for animal welfare. So thank you for joining us tonight. All right, well, thank you everybody, and thank you, Glenna, and we'll probably see you again, and hopefully we'll have another webinar with you again in the future.
Sounds good. Thanks everyone.
Okay, bye, Glenna.
Thanks for organizing, Zoe.
You're welcome. Thank you, everybody, and I'll be this is being recorded, and we hope to have it up on the BC Goat website once we get it uploaded to YouTube, which takes a few days because it's normally quite a big file. All right, well, thank you everybody, and we'll see you again soon. Bye.